Assalamu alaikum everyone. Welcome to next time. مثل ما كانت نجيب the last session, the last session of the last so we can start quickly about our presentation. We can start with the idea of the 75. I don't know if they're here. So before we start, we have to wait a little bit until to finish. We start. Okay?
Hello everyone. LCMS-based metabolomics to profile data in vitro seedlings. So we have to
So we can start so we can start with the idea number 75. The number is here now. Idea number 75.
Statistic analysis, the statistical processing of the data collected during the study is done by the software that and the data. Data of social and starting by the algorithm. The graph below shows the cost evolution of the total member in the water body over the winter season 2015 to 2016 and 2016 to 2017. It expresses the temporal distribution divided into three periods. The first period is summer and last and summer, with the next other than in general, some universities that there are shows like
Thank you, this is Bushara.
Thank you,
Thank you, Bushara, for the presentation and this show your results. And about the sound, sometimes it's bad, sometimes it's quiet, with a difficult conversation, I think. So we can ask the moderators or the audience if there is some question about these topics, please. Any question? If there is no question, so we can start for another presentation and idea of something like
Hello, let me first introduce myself. I'm EnvironmentalBiotechnologyand, PhD student at the University of Malaysia and at the National University of Ireland in Galway. I would like to thank you for giving me this opportunity to participate in this conference. So today I'm going to talk about Reptile Biodiversity in Sushmasa National Park. So we have organized this talk in the following way, starting by introduction, followed by some theory, then lectures, followed by results and discussion, and lastly conclusion. Let's start by a brief introduction about Sushmasa National Park, which is Morocco's first coastal national park, created to preserve the high diversity of its continental and marine environments. Takes its name from the Sushmasa rivers, which host two Ramsar sites of international importance. So far, very important images in the Sushmasa National Park websites, and available data remains very insufficient. Early literature shows that the first reptile records in the Sushmasa region were made in 1959 and 1962. These records were then followed by anecdotal imagery on what was previously called the National Park Project in 1988. 30 years after establishing the Sushmasa National Park, no further ornithological imageries have been made. The main goal of this work is to provide the first reptile imagery of the Sushmasa National Park since its establishment in 1991. The study was conducted by Sushmasa National Park, created along the Volcano Plot of the Sushmasa region. It covers an area of 33,800 hectares and its range over 17 kilometers. It has a dry climate and it can be divided into two parts. A northern part characterized by a hot conditions semi-arid climate and a southern part with a hot desert climate. Over the period from 2019 to 2020, several surveys were carried out, focusing on the journey prioritization regions at 30 different sites. In order to synthesize the efforts, the time extreme visual unit was used. This team involves an active visual search for species in a given area for a predefined ecological cycle. Both direct and nocturnal surveys were carried out during our most sites were surveyed at least once per season, with a total assessment area of 300 bushhours. We combined our survey data with some additional observations recorded during random encounters or provided by Sushmasa survey. researchers. However, additional observations were only added to the enrichment tree when they collected multiple vouchers and survival conditions of business in the study. In addition, we tried to determine the presence of some reptiles based on geographical problems. Sample effort of 300 prescribers. We combined our study data with some additional observations recorded during random encounters or provided by Sushmasa researchers. However, additional observations were only added to the current inventory when they included multiple vouchers and survival conditions of business in the study area. In addition, we tried to determine the presence of some plants based on geographical drugs, complexes, and chasing. For each capture, we measured the total effect made since each class. Specimens were captured and their efficiency improvement usually by rock evaluation or more forces and violence distribution. And factor inversion agreements provide authorities. It's involved to this analysis in reaching environmental members and inventory agreements of the entire structure of the contribution. By constructing sample-based simulation curves, by non-parameter researchers, Q1, Q2, Dynamic 1, and Dynamic 2 were calculated using estimated software in order to estimate total species ratio. We obtained an inventory comprising 23 species spanning across 15 families and 19 years. This includes 4 species of Biotechnology and 19 species of squid, which represents the majority of populations. We will now take a look at these species, starting by the order of unions, which includes 1 species of tortoise, 1 species of turtles, and 2 species of sea turtles. It's now turned to squid, which are represented in our area by 3 species of geckos, 4 species of snakes, 1 species of rural lizard, 3 species of true lizards, 1 species of macaque, and 1 species of cricket. We will now turn our attention to the food community, commonly known as snakes, which is represented by 6 species, including 106 of the genus Pairwise, and 5 different species. 3 of them are harmless and belong to the 3 different genera, namely macrovertical, macro, and zombies, while the 2 other species are highly venomous and belong to the genera Untitled. The observed species include Chico and the researchers are those 2 species used in designing using the binding strain now trying to compare this historical imagery with the nice tasting imagery of the last session project, which was supposed to cover 48,000 hectares compared to 53,000 hectares for the actual project. The study made by Reptile in 1988 provided a total of 18 species. However, 6 species in the previous genus have either reduced their genus or experienced systematic modifications, which led to the extinction of new species, while 3 species were absent in our new imagery. Geochemistry is symptoms and polymer are now they are not included the general as the human description. According to genetic and morphological analysis made by previous studies, soil patterns mortality, population response, and probably non-nutrition have been revised to new species descriptions and which notice are represented by soil patterns versus complex species. On the other hand, approximately 70 were not found over the course of the study. During this yes, much less the majority were, however, recorded for the first time in study here. Despite its also experience, Sushmasa farming contains nearly 20% of all marine and marine turtles, including forced endemic species. In terms of conservation concern, 5 species are more recently reptiles, owls, and the biodiversity areas, while 2 species are classified as near threatened, which underlines the importance of protected areas for those species. Thank you for your attention.
Thank you for this presentation. So, any question? Do you have any question about this topic? Can you also about the sound, I think it's open. So, if you want to repeat this video presentation, so yeah, we can repeat that. Okay, so we have to repeat this presentation with video quality. If that was okay, so wait a minute to that. Okay. Yes, after the
Well, I'm just wondering if there are any questions regarding the presentation.
Yeah, I don't know, but the most obvious is the video and the presentation. So I'm going to present again this presentation with quality. Okay? So I'm going to present again this presentation. If you have any question about the RFS query, so we can say we are listening to you.
Hello everyone, let me first introduce myself. I'm EnvironmentalBiotechnologyand, a PhD student at the Ibn Zor University of Agadir and at the National University of Ireland in Galway. I would like to thank you for giving me this opportunity to participate in this conference. So today we are going to talk about reptile biodiversity in Sushmasa National Park. To do so, we have organized this talk in the following way, starting by introduction, followed by study area, then material and methods, followed by results and discussion, and lastly conclusion. Let's start by a brief introduction about Sushmasa National Park, which is Morocco's first coastal national park, created to preserve the high diversity of its continental and marine environments. It takes its name from the Sus and Massa rivers, which host two Ramsar sites of international importance. So far, very little work has been done to study Sushmasa National Park reptiles, and available data remains very insufficient. Early literature shows that the first reptile records in the Sushmasa region were made in 1959 and 1972. These records were then followed by a herpetological inventory of what was previously called Massa National Park Project in 1988. 30 years after establishing the Sushmasa National Park, no further herpetological inventories have been made. The main goal of this work is to provide the first reptile inventory of the Sushmasa National Park since its establishment in 1991. The study was conducted at Sushmasa National Park, located along the Moroccan Atlantic coast of the Sushmasa region. It covers an area of 33,800 hectares and it stretches over 70 kilometers. It has a dry climate and it can be divided into two parts. A northern part characterized by a hot semi-arid climate and a southern part with a hot desert climate. Over the period from 2019 to 2020, several field surveys were carried out, focusing on determining reptile species richness at 30 different sites. In order to standardize certain efforts, the time-constrained visual encounter surveys was used. This technique involves an active visual search for species in a given area for a predefined amount of time, Both diurnal and nocturnal surveys were carried out during our work. Most sites were surveyed at least once per season, with a total sampling effort of 300 person-hours. We combined our survey data with some additional observations recorded during random encounters or provided by Sushmasa National Park researchers. However, additional observations were only added to the current inventory when they included photo vouchers as reliable evidence of presence in the study area. In addition, we tried to determine the presence of some reptiles based on their fecal droppings, tracks, carcasses, and shed skin. For each capture, we measured snout length, weight, sex, and age class. Specimens were captured under a scientific permit issued by the Moroccan High Commission for Water and Forests and the Fight Against Desertification, and under an internship agreement provided by Sushmasa National Park authorities. Let's move on to this analysis, in which we evaluated sampling effort and inventory completeness of the time-constrained visual encounter survey technique. By constructing sample-based accumulation curves, while the non-parametric estimators K1, K2, Jackknife 1, and Jackknife 2 were calculated using estimate-est software in order to estimate total species richness. We obtained an inventory comprising 23 species, spanning across 15 families and 19 genera. This includes 4 species of chelonians and 19 species of squamates, which represents the majority of observations. We will now take a look at these species, starting by the order of chelonians, which includes 1 species of tortoise, 1 species of terrapins, and 2 species of sea turtles. Let's now turn to squamates, which are represented in our study area by 3 species of geckos, 4 species of skinks, 1 species of worm lizard, 3 species of drill lizards, 1 species of agama, and 1 species of chameleon. We will now turn our attention to the group Ophidia, commonly known as snakes, which is represented by 6 species, including 1 non-venomous snake of the genus Hemorrhois and 5 venomous species. 3 of them are harmless and belong to 3 different genera, namely Macropertodon, Malpodon, and Psamophis, while the 2 other snakes are highly venomous and belong to the genera Naja and Taboya. The observed species accumulation curve appeared to be nearing an asymptote at 22 species, indicating that the sampling using the time-constrained visual encounter surveys was effective, and that the current inventory of Sushmasa National Park reptile is near completion. The non-parametric estimators predicted a total richness ranging between 22 and 25 species, with an inventory completeness ranging between 88 and 96%. We will now try to compare the present reptile inventory with the 1988 inventory of the Massa National Park project, which was supposed to cover 48,000 hectares compared to 33,800 hectares for the actual national park. The study made by Milado et al. In 1988 provided a checklist with a total of 22 species. However, 6 species within the previous checklist have either changed their genus or experienced systematic modifications which led to the description of new species, while 3 species were absent in our new inventory. Jiconia chalzalier, Sphenopsiformis, and Colibor hippocrepus are now included. They are now included within the genera Tarantula, Chalcides, and Hemorrhois, respectively. According to genetic and morphological analysis made by previous studies, Saurodactylus mortanicus, Acontodactylus pardalis, and Macroprotodon cuculatus have been revised to new species descriptions, and in the region, including Sushmasa National Park, are represented by Saurodactylus prostii complex, Acontodactylus margaritae, and Macroprotodon bovis, respectively. On the other hand, Acontodactylus erythrourus and Natrix mora were not found over the course of our study. Dermochelis coriaceae, Najahaji, and Taboya mauritanica were, however, recorded for the first time in the study area. Despite its small surface area, Sushmasa National Park contains nearly 20% of all known Moroccan terrestrial and marine reptiles, including 4 endemic species. In terms of conservation concern, 5 species among the Sushmasa National Park reptiles are classified as vulnerable on the IUCN Red List, while 2 species are classified as near threatened, which underlines the importance of protected areas for those species. And thank you for your attention.
Thank you very much. I think it's better now we're trying to video. So, you have any questions? Any questions? From the audience? So, if there is no question,
Thank you very much, for this presentation. The importance is that.
Okay. So, we can start with the
87, page 87. If the author is political. I think the area number 86.
In like 10 minutes, I will be talking about using LCMS-based metabolomics to profile date palm in vitro seedlings. So we have 2 key elements. We have 3 date palm cultivars and metabolomics, which is a widely used approach in fingerprinting. So we will try to establish a metabolomic fingerprint of the seedlings, then we will compare them to each other, and finally we will have a look on the more significantly enriched metabolic pathways. So I will skip all the generalities about date palm, which is a well-known plant, and talk directly about in-targeted metabolomics. Well, the goal of metabolomic profiling is to obtain the maximum amount of data on metabolites. So we can be able to have biomarkers to identify environmental or genetic distribution, which is the case of our study. For targeted metabolomics, the purpose is to extract all the metabolites, known and unknown, and this is by using a high-performance tool. In our case, it is a high-performance liquid chromatography coupled with mass spectrometry in positive and negative electrospray ionization modes. For the methodology, we started with an in vitro germination of 3 date palm cultivars, which are Deglet, Noor, Ghars, and Meshdegla. Then we collected the samples by snap freezing them in liquid nitrogen, which is a critical step. After that, samples are aliquoted and prepared based on the work of Salim et al. 2016. Next, the raw data is obtained thanks to this couple, as you can see in the picture, UPLC with Q-exactive mass spectrometry. After that, raw data were processed in XCMS, and the results matrix obtained were analyzed on Metabolomics 5.0, which is, by the way, a very useful tool to perform statistical and data visualization. For this presentation, I will just compare the roots in both modes, positive and negative. Before getting to the results, first I would like to talk about normalization, which is an important step in data omics analysis. As you can see, when data are normalized to median and log transformed, they become two-tailed, or like we usually say, they respect a normal distribution. This makes all the data accurately comparable to each other and would allow us to perform all the parametric analysis. Now let's get to the results. Here you can see a presence-absence comparison of the detected metabolites, which is, by the way, a naive and, let's say, a neglected approach in statistical analysis. But we will see that it is justified because it would give us the same conclusion as would the next statistical analysis do. Well, as you can see in positive mode, 91 metabolites were detected exclusively in the Deglet-Noor, and we have 115 in negative ionization. On the other hand, Ghars and Meshdegla have more in common detected metabolites with each other than with Deglet-Noor. And I would like to point out that the filtration of the metabolites was done manually. We were pretty severe in removing all the metabolites that were not detected in absolutely all the samples of the same group. The analysis of variance or ANOVA revealed more than 500 significant metabolites. We have 521 metabolites in positive mode and 509 significant changes in the negative one. And when I say significant changes, I mean really significant because the data filtration was somehow done severely. And that was by using FDR p-value correction that eliminates all the false positive and the false negatives. Also, the p-value of significance is set to 0.01. Here, as you can see, the correlation heat map using Pearson-R correlation gave us 3 major clusters in both positive and negative ionizations. What is interesting is the patterns that we can see in the negative mode, where there are metabolites that are more abundant in Deglet-Noor than in the 2 others, and the opposite. The result is a negative correlation between these features. But there are some features in the center of the heat map, as you can see, that are low in Deglet-Noor and high in the 2 other cultivars, but do not counter-correlate the exact opposite pattern. Now we will see what other multivariate analysis could reveal. Pci analysis, or principal component analysis, shows that Noor is different from Ghars and Meshdegla, which are more or less similar, which is reinforced by the hierarchical clustering, where we used Pearson distance measure and mean-based clustering algorithm. On the other hand, partial least squares discriminant analysis, or simply we call it PLSDA, there are 2 important measures. One is the variable importance in projection, VIP, and the other is the weighted sum of the absolute regression. Metabolites are represented by the M over Z and the retention sign. The colored boxes on the right indicate the relative concentration of the corresponding metabolites in each cultivar. This analysis is as indicated discriminant, where we can see that the three cultivars are more separated than in the PCA. On the right, we have an analysis type which is even more discriminant. It is called sparse PLSDA or simply SPLSDA. This algorithm can reduce the number of variables, in our case metabolites, to produce robust and easily interpreted models. We can see here that for the positive ionization we have the same logical sequence. The principal component analysis revealed the same pattern, where diclofenac is distinctly different from the two other cultivars. But with the discriminant analysis, PLSDA and SPLSDA, by reducing the number of variables, we have an obvious distinction between all the groups. For the SPLSDA, it seems like the dominant pattern is when GARS has the highest relative intensity of metabolites, Mejdegla the lowest, and diclofenac in between. We'll proceed to the pairwise comparisons. Here you can see the Volcano Plot comparing diclofenac to GARS. Volcano Plot is one of the most representative graphics for omics analysis. Here we can see the significance based on the p-value from the t-test combined with the fold change ratio. Both parameters are required to determine if a metabolite changed significantly or not, as usually said, upregulated or downregulated. In this comparison, we set 2 as a fold change threshold and a p-value of 0.01. In addition, we can see clearly that the FDR correction has a huge impact on the results. By applying the FDR correction, we have 258 upregulations and 228 down significant regulations. But when we remove the FDR correction, 177 false significant changes are removed. Price comparison of all the cultivar is more obvious when having the top 1000 features. The relative intensity heat maps shows a sort of a contrasted difference between diclofenac and GARS, diclofenac and Mejdegla, but not between GARS and Mejdegla. The same contrasted difference is even more clear by visualizing the correlation heat maps, where a huge positive correlation is present with an analog negative high correlation, which is not the case between GARS and Mejdegla. Now let's see the fingerprint of the cultivar roots. Here we can see the heat maps of all the detected features in both positive and negative ionizations. The average of relative intensity is represented with red for the highest values and green for the lowest. These two heat maps are the proof that metabolomics are a tangible approach to identify the genetic differences. And finally, we go to the enrichment analysis of the metabolic pathways. As represented in here, the main differences between diclofenac and the two other cultivars are those where amino acids metabolism are affected, like tyrosine metabolism, phenylalanine metabolism, tryptophan, arginine, and cyanoamino acids metabolism. Also, we have secondary metabolites biosynthesis which are affected, but the significant changed metabolites are small compared with the total detected ones. And here again, the difference between GARS and Mejdegla is slightly remarkable, with only 4 enriched pathways. And to conclude, we can say that this study showed us that metabolomics can be a revelatory method to explore genetic variability. Also, results reveal an interesting singularity of diclofenac and a curious similarity between GARS and Mejdegla. Thank you.
Thank you very much. We really enjoyed this presentation.
For this important research,
About the date. So, it's very important with us. So, this question of this please. So, if there's
we've lost another presentation.
yeah. So we have to think again on this topic, start with this and we can just do another presentation and the idea of 87
Or 98.
So we can start with the idea and what?
98.
98. Okay. 98. So the 98.
If the authors is basically on multiple.
Everybody, I am Quadrия Sara. I'm a PhD student from the University of Shadli bin Jid al-Taraf. I'm talking about part of my PhD, that is stock evaluation of the European hake on the East Algerian coast. European hake is one of the most important demersal species of Mediterranean demersal ecosystem. This species of great commercial value show growth overfishing, which catches basically focused on juveniles. European hake is generally found on muddy bottoms and distributed on a wide depth range from the coastline down to the depth of 1,000 meters. Is one of the most heavily exploited fish species. European hake has been reported as one of the most important species, which make it economically interesting. This explains the choice of our species. The generic name of European hake includes 14 species of marine fish whose taxonomic classification. In this study, we asked to evaluate the growth parameters of the European hake in the Algerian eastern coast, which is an important indicator for the sustainable management of this species. The study area is located in the extreme east of the Algerian coastline, between Staibi in the west and Al-Qala in the east. We have successively the Bay of Staibi, the Gulf of Hannaba, and coastline of Al-Qala, and this is location of studied seat. We collected monthly during a period from April to June 2021. Biometric parameters was measured. That is, length total, length standard, weight total, and weight eviscerated. We examined 121 individuals in this study, and we used the VISA tool to determine the parameters of the von Bertalanffy model. This graphic represents the size frequency classes of captured fish. The total length varied between 11 and 34 centimeters. The size frequency distribution revealed that size classes of 17 to 19 centimeter individual dominated. This graphic represents weight frequency classes of captured fish, whereas the total weight was between 8 and 440 grams with the mean for 20 centimeter and 68 grams respectively. Concerning the sex ratio of the individual sampled, 55% were considered female, 22% male, and 23% indeterminate. Age determination by the indirect method Bhattacharya. The method of Bhattacharya facilitated to group the sample of European hake into 4 cohorts. The value of size by age and the number of individuals in each age classes are presented in the table. Growth parameters. The Van-Bertalanffy growth parameters were estimated on using elephant routine by FISA-2 program. For the asymptotic length, the growth coefficient, and the theoretical age and the performance index was present in this table. This graphic represents the length and the weight relationship, and the value of B reflected a positive allometry. As the result of this study, we have demonstrated that the European hake is among the demersal species with high commercial value, and one of the most important fishery resources in Mediterranean Sea, and particularly in Algeria, where this species is exploited all the coast. Thank you for your attention.
Thank you, Sarah, for your presentation. Yeah, Rania,
Salam, what about number ED 86?
86 for Sarah.
Yes, listen, I have a problem of connection.
Yeah, so if you have another question, you can say it here.
I have problem of connection.